Welcome back to day 2 of the Spatial Biology Revolution. We're excited to have you here and we can't wait to see the incredible lineup of talks we have in store for you today. My name is Princess Senate, conference producer at Technology Networks, and I am pleased to welcome our opening talk today brought to you by Dr. Ninning Liu. Light-Seq, Light-Directed Spatial Indexing and Sequencing. Ninning Liu is a staff scientist in the Molecular Robotics Initiative, where his focus is on engineering DNA-based tools for spatial omics, biosensing, and high-throughput barcoding. His current interests include working at the interface between imaging and sequencing technologies, applied machine learning, and exploring application areas of translational technologies at the Wyss Institute. A warm welcome to you, Dr. Liu. Following the presentation, we will have a Q&A session, and we welcome any questions that you may have. Please ask questions using the Q&A system to the right-hand side of the video player. I will now hand over to Dr. Liu to begin his presentation.
Thank you very much, Princess, and thank you for this opportunity to allow me to talk about our Light-Seq light-directed spatial transcriptomics platform. So Light-Seq itself has been published for a little over a year now, and hopefully I'll talk a little bit about how it works, and you can decide whether or not it can fit into your workflows. So I'd like to start by motivating this talk by calling your attention to two particular numbers that I like to think about when thinking about spatial transcriptomics. So the first number is 10. And 10 is roughly the size of a single cell. And in tissue, I mean, this size can range anywhere from 1 to 10 microns. And so on the left side, this is an overview of a mouse retina tissue section. And if we kind of zoom in a little bit, the blue is the dapiscene of all the various cells in this mouse retina tissue section, and the orange is a protein stain for one particular cell type, a dopoamerogenic amacrine cell. And as we slowly zoom into this one cell type, the orange amacrine cell is actually about 10 microns in diameter. So it's actually quite big compared to the other cell types in the mouse retina, which is probably closer to 1 or 2 microns. The other number that I would like to call your attention to is 63,494. And this is, well, last time I checked, when the complete sequence of the human genome was published, this is the number of entries in the human genome that are labeled as a gene. And with these two numbers, you can kind of get the sense of how difficult it is in spatial transcriptomics to fully profile the gene expression profile of a single cell. So, I mean, within a cell, you can have a wide number of permutations of different gene expression patterns. Zooming out a bit to the full mouse retina tissue section. So this is a protein stain that our collaborator Emma did, and it very beautifully highlights how biology is organized across different scales of space. And each of these colors here is a protein stain for a particular cell type, and you can see that each individual cell type are stratified into distinct layers. And with Light-Seq, what we like to do is use light to parse out each individual layer for next generation sequencing. And if there's a particular region of interest that you want to target, you would use light to parse it out, deliver a DNA barcode to that region, a DNA barcode that targets the full transcriptome of that region of interest, and then recover that information with next generation sequencing. So if you ask me to summarize the vision of Light-Seq in one word, I would probably say Wyss, which is a bit cheating, but it stands for, what you select is what you sequence. And which is, I'm attempting to summarize in this slide, which is you can use light to directly target regions of interest for next generation sequencing. And there are quite a few advantages with using light. I mean, one is it is very penetrate into your sample, and you can change the size of the ROI down to potentially the diffraction limit. So in addition to targeting whole regions of the tissue for next generation sequencing or differential gene expression, you could potentially shrink the size of your ROI down to a single cell for single cell transcriptomics, which I'll actually talk about a little bit later. And in addition to that, because light is generally non-destructive to the sample, you can follow it up with some immunostains or some other downstream assay that you're interested in. Now, I don't really need to go into this with this audience, but spatial transcriptomics is obviously a very exciting field, and that's kind of reflected in the large diversity of technologies that many other authors have put out. So including Light-Seq, which is a 2D array, MERFISH, Wyss-Seq, I think published in 2015, and then there are other technology platforms that are, in terms of using light, it's probably more similar to Light-Seq, which is laser capture microdissection or use of a photocleavable spacer. The main difference is with Light-Seq, what we're trying to do is deliver a single DNA barcode to the whole transcriptome, whereas something like a photocleavable spacer would release a library set of barcodes that you would extract and collect for next generation sequencing. And with that overview, I'd like to dive into probably one of the more powerful applications of Light-Seq, which is single cell transcriptomics, but not just single cell transcriptomics, but rare cell single cell transcriptomics. And traditionally, at least when spatial transcriptomics was first being invented and put out, it has been rather difficult to get the resolution down to the single cell. But given the rapid pace of development, I think that quite a few technology platforms have achieved single cell resolution. The layer further than that is there are some cell types that are extremely rare in your biological sample. And this has also been traditionally very hard to isolate just because of how interspersed and sparse that they are. One particular cell type that we decided to pick for Light-Seq was a cell called dopoamergenic amacrine cells, or DACs for short. And these are probably some of the rarest cells in the mouse retina and possibly the human retina as well. And in this particular tissue section, we have about 6 of these cells, and any single section might only have about 5 or 10 of these cells, with a single retina might only have about 100 of these cells. So that kind of gives you a sense of how rare these cells are. What you're seeing right here, the kind of the orange stain, is how we identify these cell types, which is with a protein stain that targets a known marker for these cells, which is for tyrosine hydroxylase. So I'll show a video that my co-authors Emma and Josie made, illustrating how we use Light-Seq in order to parse out the differential gene expression for these cell types. So you can see we identified these 6 cells in this instance with our TH plus protein stain. And then we barcoded those 6 cells with one particular barcode. We'll revisit that later. And as we zoom into one of these particular cell types, the orange is, you can see the cell body of these amacrine cells in orange, and we applied 2 different barcodes. So the magenta barcode is to the main cell body itself, and then the green barcode is to the adjacent cells of that TH positive amacrine cell. So the adjacent cells are also considered to be amacrine cells, they just happen to be TH minus. And once we applied these 2 barcodes, we can then extract the cDNA library from these 2 regions and then compare the differential gene expression between the TH plus and TH minus cells. And what you can see here is we actually did pull out several genes that were enriched in the TH positive DAC cells, including the known markers, of TH and PT for the Th positive amacrine cells. In addition to that, we actually also pulled out several markers that, to our knowledge, was not previously characterized in the literature, or at least was not very well characterized in the literature. And our collaborator, Emma, went back and validated some of these novel markers that we pulled out with a FISH assay. And you can kind of see on the right side of these puncta that shows up in the amacrine cells of the gene targets that we identified. So I hope that gives people a sense of what you can do with Light-Seq. You can select an ROI all the way down to a single cell region, and then potentially get single cell transcriptomics from a sample of even just a few cells in your tissue sample. But to go into what's the technology behind Light-Seq, to boil it down, it's really just the use of spatial light modulators to deliver a DNA barcode that has a photoactivatable element. In our case, we use a modified base called CMVK, and I'll go into a little bit detail later about how it works. But this base modification is what we use UV light to photo crosslink and deliver that barcode to a region of interest. On the left side that you see here, we use a spatial light modulator called a digital micromirror device, or DMD for short. And people in the optogenetics field are probably more familiar with this. It's been in use in optogenetics for, 10 to 15 years or so. But it's actually quite an old technology. It's been around since, I think, the 80s. It's been used in overhead projectors, or in this case, they used it for a digital cinema projector. And what it does is it has millions of these micron-sized mirrors, and you can generate and project an arbitrary light photo mask onto any surface or sample. What I'm showing here in this video is an example of what you would see if you use the DMD device with your microscope. So it just comes as a microscope attachment. All you have to do is literally just draw the region of interest with your mouse, and then the DMD device will interpret that region and then flip corresponding mirrors to project a cross-linking light, in our case UV, onto the sample area. So to demonstrate the iterative cross-linking that we can do with something like a DMD, what I'm showing on the right side right here is we generated three photo masks, and we barcoded in sequence. So what we do is we introduce a fluorescent DNA barcode, turn on the UV, wash out all of the unbarcoded, non-cross-linked DNA strands, and then repeat the process for 2 and 3. And then once you overlay all of these strands, you can get a 3-color Penrose triangle. For those of you who are curious, the one that we used, the DMD that we got from Mitex, but there's other vendors out there that for your microscopes, if you're curious. Now, it's not just a solid image that you can use with the DMD. You can actually project any arbitrary image onto a surface. And to test this, we actually got an image of a cat. So this is actually Josie's cat, if you're curious. And you can take this image and then convert it to a binary image. You can do this in Photoshop. So with this image, you convert it to a binary image like so. And this image is literally just a image with 0 and 1 pixels. That's all it is. And the DMD itself will be able to interpret all of the white pixels as the location in which to turn on or flip on the corresponding mirror corresponding to that location. And then we cross-link the fluorescent DNA onto the surface, and then recover the image of the cat that you see here. So actually with this image of the cat, you can actually start to see the resolution and the resolution limits of the DMD. So, this experiment does serve a purpose. So each of these individual spots that you see, like with this cat eye, like you can resolve it quite well in the fluorescent image. But once you start to cluster these spots very close together, so if you look at, I don't know, one of the whiskers, some of the spots are very close together in the binary image, and then because of diffracted light or scattered UV light, the cross-linked fluorescent image actually looks a little bit more blurry. Okay, so that's the technology or the equipment required for Light-Seq. In addition to that, there are two main components for Light-Seq in order to perform the experiment. So the first is a fast spatially prescribed photo cross-linking platform. And with the spatial light modulator, we generate a photo mask to project an arbitrary pattern onto a surface. The photochemistry that we use is something called 3-cyanovinyl carbazole, or CMVK for short. And it is a commercially available DNA-based modification. Like, we got ours from GeneLink. And it is unique in that you can use, this base modification, what it does is that it can form an interstrand crosslink with a complementary DNA strand. So if this strand has the CMVK-based modification, it'll form a crosslink with this complementary strand here. And it's also useful in that it can go to completion in about 1 second of UV exposure. So this particular base modification, it's not as well known or commonly used, I think, as something like a photocleavable spacer. But these two actually serve very complementary purposes, in my opinion. So with the photocleavable spacer, you can release a DNA barcode, whereas in this case, you can use this modification to deliver a DNA barcode. And one of the first papers that we published using this particular base modification was a super resolution labeling strategy, which we called Action Paint. So we targeted like three specific regions on a DNA origami, and then took an image to confirm. So, and the second part of the second component that Light-Seq requires is a one-step DNA barcode stitching reaction. And what I mean by that is we deliver and crosslink a DNA barcode to our cDNA library. However, what we would like at the end is a single contiguous DNA sequence that has our cDNA sequence on one side, and our DNA barcode sequence on the 5' end, that which we can use and recover with next generation sequencing. So for the DNA barcode stitching reaction, I'll go into a little bit detail about how that works. Is if you imagine on this strand, we have our cDNA sequence that we need to recover and sequence. And on this side is our DNA barcode sequence that we want to stitch it together. What we do is we have this cross-junction geometry, and we introduce a polymerase, in a strand displacing polymerase in this case, to copy the cDNA sequence up until the cross-linked base. So imagine a polymerase opens up at that little base pair hairpin region, copies up to the cross-linked base, and because it is a covalent interaction, the polymerase should not be able to address this area. And eventually, because it can't progress forward, eventually the polymerase will fall off. The strand design that I'm showing here with the P or P star, these are self-complementary regions. So P is complementary to P star. Ultimately, what this does is it facilitates a three-way branch migration. So you can think of it that way if this geometry looks a little bit unfamiliar to you. So with this three-way branch migration, eventually this extended strand will be able to base pair with this region labeled P star right here, and that will allow a polymerase to once again come in and copy and extend this region in order to create a fully contiguous DNA barcode plus cDNA region. Okay. In our supplemental figures, we actually had an in-depth view of one example of this cross junction. So we have some primary domains we use for PCR amplification. If you're curious, the naming convention was an iteration from a previous paper of one of our co-authors. So he had a GAT-A primer, we use GAT-C, GAT-E. And we have our CMYK-based modification here. So this is the cross-link region that the polymerase should not be able to cross. And then we have our cross-junction region right here. In addition to that, we have a UMI for to remove PCR duplicates, as well as our barcode sequence. So for the workflow of Light-Seq, if what we do is for our sample, we do start with a fixed and permeabilized sample. So that is important to take into consideration if you're thinking about applying it in your system. We've tested, mostly paraformaldehyde, and the paraformaldehyde was the one that we tested the most extensively. But in principle, methanol should also work, but we don't have as much experience or data for those two conditions. And what we do with our sample is we generate our cDNA library with an in situ reverse transcription. So we introduce a reverse transcriptase, generate the cDNA library with a set of degenerate primers. And what we introduce and cross-link the barcode to is an overhang on the cDNA that's shown right here. So and so this barcode, crosslink, and cDNA DNA set that I'm showing here, that is the cross-junction region that I described earlier. After we barcode our cDNA, we do an RNase H extraction. So we get rid of the endogenous RNA to help release our cDNA barcode complex. So unfortunately, in our current iteration of the workflow, we can't do a follow-up FISH assay on the same sample. Once we have our set of cDNA and library, that's when we do our cross-junction synthesis, and that's when, and then by the end of PCR amplification, what we should have is a set of double-stranded DNA that has both your barcode and cDNA attached, which you can then go on to do next-gen library sequencing prep. And finally, for our barcoding workflow, when we tested our barcoding workflow out, one of the first experiments that we did was we started a cell culture with a mixture of both mouse and human cells. And then we targeted the mouse cell with one barcode, the human cell with the other barcode. And what I'm showing here is a large overview image with cyan and magenta as the two cell types that we targeted within a single well. And as we zoom into this region, the workflow is essentially you just draw a ROI with your mouse around the cells of interest. So the human cells actually had a GFP to help identify them out, and the mouse cells were fibroblasts, so they were actually quite easy to identify. And we targeted them with the two different barcodes, which is what you can see here. For cyan and magenta. And then after extracting the sequences out, you can then do next generation sequencing and map the sequences to mouse and human reads. And because the samples themselves were still intact after this process, you can follow it up with a protein stain. So we had some antibodies targeting some very common protein markers for both of these cell types. Right now, our DNA analysis pipeline is pretty basic. We're just looking at expressed genes in both mouse and human cells. And and the pipeline itself is fairly standard across for differential gene expansion. So getting into some of the sequencing results that we got for the mouse retina tissue section. So I showed this image earlier of the 3 regions that we targeted in the mouse retina. After we did a proof of concept on these mouse-human cell mixture samples, we had a collaborator come into the project, Emma, who works in Connie Sepko's lab, so that we can really get a sense of how well Light-Seq can work in a tissue sample that probably has lots of heterogeneous cell types mixed together. Although in this case, the retina might be the ideal case because all the cells are stratified into very nice layers that you can target. So we targeted 3 specific layers in the mouse retina. So the first layer was the magenta, the outer nuclear layer, the bipolar cell layer, and the ganglion cell layer, shown in magenta, cyan, and green respectively. And then when we pulled out And then when we pulled out all the genes that are expressed in this region, we found thousands of differentially expressed markers for all of these 3 regions that we targeted with these pairwise volcano plots, if you have you, that I'm showing right here. Now, some of the markers that Emma went back and highlighted here, these are markers that are well known to be expressed in the region of interest. So, for example, I think RP1 is rhodopsin. No, Rho is rhodopsin protein, which is known to be expressed heavily in the outer nuclear layer. And so I'm just summarizing the retina results here. Once again, we barcoded the 3 particular regions, and then we followed up with a protein stain that I showed in the earlier title slide for, again, some well-known protein markers indicative of the regions of interest. And in addition to that, we also showed some of how the reads drop off for each of the replicates that we did. So one of the things that we actually didn't do, unfortunately didn't get a chance to do in the paper, is ribosomal depletion, because as we started looking at the sequencing data, we ended up finding that almost half of our reads, if we're just looking at the exon reads, almost half of it was taken up by ribosomal RNA. And with some later experiments that I did, which unfortunately didn't make it into the paper, like if you do a ribosomal depletion, you can actually reduce this by almost a factor of 5. So if you're interested in rare gene detection, that's obviously the way to go. And finally, to summarize the rare cell transcriptomics, so the rare cell transcriptome was really enabled by the fact that we could do an immunostain in addition to Light-Seq protocol on the same sample. And I'm showing the protocol that we did here. We actually kind of switched the order of operation. So in the cell mixing experiment I showed earlier, what we did was we did the sequencing first, and then we followed up with a protein stain. In this case, for rare cell transcriptomics, we actually had to identify the cells first with a protein stain. So the first step was the immunostain. Then we followed it up with a DNA barcode, workflow. Now, so up until this point, what we did was we really just identified the cell types of interest and the regions of interest in bright field, like morphologically. And for most cell types, or for maybe a majority of cell types, you can probably get away with that if you're very familiar with how the cells look like. However, for these cells, when I was looking at them under bright field, it was borderline impossible to identify them just from bright field alone. So this is an instance where you really have to combine an immunostain to target and help identify, and use that immunostain to guide your DNA barcoding regions. So in the paper, we showed this figure where we targeted this particular cell type, the amargin cell, the adjacent cell, and then picked out the differentially expressed markers. And as I showed before, Emma went back and validated all or most of these markers with an in situ fish experiment. Okay, so that's essentially all of the results that we had in the paper. But in terms of what is in store for Light-Seq and what we can do with Light-Seq, so one thing that Light-Seq lends itself well to is you can integrate an image recognition algorithm using any off-the-shelf machine learning model that you can get from GitHub to help you identify a cell's of interest. So with the DMD, like what you can do is you can actually just upload a binary TIFF file or a segmentation mask onto the DMD and use that as your photo mask. So what I'm showing here is I just used a pre-trained model that was already published to help identify the cells by the DAPI stain, and then it created a segmentation mask that you can then use to barcode the cells of interest. Now, if there is a particular cell type that you're interested in, I mean, nowadays, I don't think it's actually all that difficult to train a new model to identify your cell of interest. In addition to that, like for ongoing work that we're doing with Light-Seq, so one question that most people are probably interested in is whether or not we can do this on FFPE tissue. Well, I mean, the short answer is yes. I mean, this isn't ongoing work, but there's obviously always a lot of caveats when you're talking about FFPE. So, you know, we're working with like freshly cut FFPE samples, which is, you know, vastly different from samples that were cut and then stored in the fridge for, I don't know, a long time. So the RNA quality is always going to be the determining factor regardless of the technology platform that you use. Secondly, we are exploring other downstream assays that we can incorporate into the Light-Seq workflow. So, and one particular sticking point for the transcriptomic and proteomic field in general is the on a global scale, at least, the transcriptome, the genes expressed, is not really that well correlated with the proteins that are present in the sample, and because the proteins in the sample are still more or less intact, you can follow it up with well, it does give you the option of using a destructive workflow like mass spec in order to identify all the proteomic space of your sample of interest. And finally, because we are using light to direct our barcode to a region of interest, in principle, it can get down to the subcellular level. So that is an open possibility. I'm showing here is a grid of dots that are about 1 micron size that you can barcode onto a glass surface. And then in addition to that, we did do some preliminary experiments where we targeted some nucleolar regions in a cell culture, which is hopefully what you can see. You can see with these magenta spots right here. So I'd like to start wrapping up my talk. If you're interested in learning a little bit more about Light-Seq, we do have a website, Light-Seq.io, that goes into detail about all the protocols and also has the GitHub link for all of the software packages that we used. If you were interested in how some of the videos that I showed earlier, that's actually a Google Maps demo that I created for the Wyss retreat a long time ago, but it's still up there, I think. So you can go to this website to browse the amacrine barcoding experiment that we did earlier. And finally, I would really like to thank all of my co-authors that helped make this project possible. So Josie and Sanem and Emma are the main authors of the paper, as well as Peng, who was my postdoc advisor at the Wyss Institute, where this project was created, and of course our funding and validation sources. The paper is online and open access. It's been out for roughly a year now, and for a disclaimer, this technology is there is ongoing commercial activity for this technology in digital biology, and I encourage you to reach out to Josie or Emma if you're interested for that. And so thank you for your attention, and I'd be very happy to take your questions.
Thank you very much, Dr. Liu. As with all the talks, we are likely going to have lots of questions from the audience. So I'll start with your first question. What is the largest barriers to entry to spatial omics field?
Yeah, so I mean, I'd say the first one would be cost. Like a lot of spatial omics, especially spatial transcriptomics, requires a lot of sequencing. And I mean, depending on what you're interested in, like each experiment can be like thousands of dollars. So I mean, there is hopefully like sequencing costs will go down in the future, and maybe that will not be as big of a barrier of entry in the future. The second one might be equipment and technical know-how, which is what we hopefully attempted to address a little bit with Light-Seq. So the equipment itself is all largely commercially available, and in principle, like all you really need is a DMD, and you can buy these like DNA modified strands, and you could do Light-Seq in your lab if you're willing. But obviously, it doesn't always work that way.
Yeah, those are some great points. It's clear there are some challenges, but there's also tremendous potential in the field. So given these hurdles, what do you think, what do you see as the next steps for spatial omics?
Yeah, so it's a difficult question to answer since it is progressing so quickly. I mean, I'd say at the end of the day, spatial omics, especially spatial transcriptomics, is answering a fundamental question, but a very basic question at the end of the day, which is, where is my gene expressed? And because of there's a lot of genes and the cell is very small, this question took a lot longer and a lot more effort to answer. But the next question that logically follows is, what is it doing? So spatial omics is just a snapshot in time. So we're still missing things like epigenetic modifications. It doesn't really capture transcriptional bursting events. So applying like a time course assay, figuring out some way to combine, like DNA methylation states and proteomic expression would probably be the next avenue for spatial omics.
Excellent. Now looking to the future, do you envision spatial omics becoming a standard assay in labs, maybe a go-to technique for researchers? How do you see that evolution?
Yeah, so I mean, my pessimistic answer is probably not, but my optimistic answer is it might be as standard as next generation sequencing has become lately. And that actually took quite a bit of time in order for that to happen. So, some labs, like a next generation sequencing instrument is not like a standard equipment in a single lab. It's usually a core. And so that's kind of what I envision spatial transcriptomics will probably be, is like, because of the instrument requirements, the cost, and the know-how that you need to operate it, it's not as simple as just PCR. So obviously some people have kind of gone down that route. They've established a spatial omics core, and they've had full-time staff and equipment dedicated to doing those assays.
Yeah, interesting. And speaking of evolution, we know that AI is making big waves in healthcare. How does spatial omics fit into the AI revolution in healthcare, in your opinion?
Yeah, I guess that's definitely the $1 million question. It's and it's also probably impossible for me to predict where this is going to end up being. I guess with anything, like, it really just depends on what you're interested in looking at. So, you can imagine using spatial omics to predict disease progression or drug response based on a patient's spatial genetic markers, right? But for something like that, you would need population level data, and like, I don't remember how long it took to establish like the Cancer Genome Atlas or all the databases for single cell sequencing, but it took quite a while. And with spatial omics, I mean, it's still a relatively new field, but I do definitely foresee, like, in the next 4 years, next 10 years probably, as we get more data and get more population level genetics, spatial transcriptomic data for thousands of patients, or ideally tens of thousands of patients, that's when you can really start leveraging all of this data for AI models.
Thank you so much, Dr. Liu, for sharing your expertise with us. Unfortunately, that's all we have time for today. Any questions that we didn't have time to get to will be sent to the speaker after the event. As with all the other talks in the event, this talk will now be available to view whenever you want for the next 30 days. That just leaves me to say thanks again, Dr. Liu, and we will be back soon with the next talk.
Thank you.